in different conditions, such as the viral infection and bacterial infection. What we are doing, we are using the PCR technique and ELISA technique, using which we can detect even a single molecule or single viral particle also. And using the rest of the techniques other than the biotechnological applications, we cannot detect the virus at initial stage, up to when the symptom is not being developed. So before the development of any of the symptom, we can use the PCR as a diagnostic procedure to detect the DNA or any of the molecule. Based on the DNA or the molecule, we can get the information about the disease. And I told you where the PCR technology has been utilized, in case of AIDS, cancer, and coronavirus infection. You know that RT-PCR we are using. So here RT-PCR is a kind of PCR we are using to detect the viral infection. Even though if a single virus is infecting your body, then also you can detect with this RT-PCR. That's why we are preferring RT-PCR the most as compared to rest of the diagnostic procedures. Now the second procedure we had discussed is ELISA, and I told you to make it remember that in which particular diseases we are using this detection technique or diagnostic procedure. And you will have to remember its full form. Okay, enzyme linked immunosorbent assay, you will have to remember the full form of ELISA, and its application you will have to remember. You just see here that mainly this ELISA, although ELISA is applicable in different diseases, but mostly we are using this ELISA in case of detection of AIDS. So in case of AIDS disease, we are preferably using this ELISA. And I told you before also that this PCR is a technique in which we are detecting the disease on DNA or RNA level, means on nucleic acid level. If we want to detect any of the abnormal thing or any of the virus or bacterial infection, then we can detect it. So PCR is based on nucleic acid detection, and ELISA is based on antigen specific detection. So in case of ELISA, we are using antibody, and in case of PCR, what we are using nucleic acid. So here, in case of PCR, PCR is based on nucleic acid molecule, and here ELISA is based on antibody and antigen interaction mechanism. So you will have to remember here that ELISA is mainly based on the interaction of antigen and antibody reaction. Okay. Now we'll come on the third procedure, using which we are detecting certain genes. If we want to check that particular gene is present in this tissue or not, we can detect it. If we want to check that this particular sequence of DNA is present in this cell or this tissue or organ or the whole animal is not, then we can do it. Okay, means what is probe? First, we will have to understand what is probe. Then only we can understand its application and its role in diagnostic procedures. So, and when we are talking about its application, then mainly probes are used to detect any of the genetic disease or sometimes in cancer also. Okay, so what we are doing, probe is a long stretch of nucleic acid, single stranded stretch of nucleic acid. Either it could be DNA or RNA. Okay, so its size ranges around 20 to 30 nucleotides. So it is a stretch of 20 to 30 nucleotide of DNA or RNA, which would be made based on its complementary nucleotide. Okay, so based on the complementary nucleotide sequence, what we are doing, we are either making DNA or RNA stretch, and the size of this stretch should be in between 20 to 30 nucleotides. So you know that if two two single stranded nucleotides are there, if they are complementary to each other, then ultimately they will bind. Okay, so suppose if I want to detect a particular stretch of DNA or RNA, then based on that we will make a synthetic stretch of DNA and RNA of the complementary of that particular DNA or RNA. So what we will do, we will allow to adhere, we will allow to bind based on the complementary region. I'll show you the figure, then you can understand its role in detection of diseases or uh, any of the disease. Suppose I want to check this sequence. Either this sequence is there in DNA or not. Okay, so this sequence. Okay, so what we will do? Suppose I have got an information that if any of the tissue, this particular sequence of DNA is there, then it is causing cancer. Suppose TAGGCT is the sequence which actually causes cancer. Suppose I have got this information. Then how I will detect that this particular sequence is there? Suppose this one is a particular mutation which causes cancer. Okay, so if I want to detect this particular site, is this particular mutation in the DNA of a particular tissue, then what I'll do, I will synthesize the complementary sequence of this particular stretch of DNA. So since we have synthesized this particular sequence of DNA, and after that we will take the tissue, the biopsy has been taken. We used to take the biopsy of the patient, and what we are doing, we are doing several diagnostic procedures. So what we will do, we will take the, suppose we are talking about the lung cancer, so we will take the tissue of the lung. After that, isolate the DNA. After that, what we will do, we know that this one is the sequence which causes the lung cancer. Then we will make the probe. So probe, it will be based on that particular mutated sequence, and it will, that due to complementary basis, we can allow to bind both of these sequences by their base pairing mechanism. So what we will do, we will take probe and we will allow to attach the probe on the DNA, or even though in the tissue we can do it. We will perform certain procedures using which we can perforate the tissue. After that, we will allow this probe to bind. If this particular sequence this probe is finding, then it will get attached. And if the sequence is not there in that particular tissue, then obviously it will not adhere. It will not bind with the base pairing mechanism. Okay, and you know that this probe will also have some signal sequences. Okay, so after that, what we will do, we will detect the signal. So suppose we have taken the tissue sample, or to which we are going to check that there is a particular mutation or not. So suppose we have taken the tissue. After that, we will allow the probe to bind with this the DNA present in the tissue. After that, we will do the washing procedure. If this probe will get its complementary sequence, means if it gets the mutated sequence in the DNA, then it will bind with that. After washing procedure, it will not be removed off. But if this probe is not getting its complementary sequence, then it will not bind. So what will happen in positive sequence? If the patient is having that particular mutation, then what will happen? The probe will bind that with that sequence. Probe will bind with the DNA. After that, since we have attached some signal molecule on the probe, then this particular tissue will have that signal molecule. And when we will do the further procedure, then we can detect by different way the signal molecule will give us the signal that this particular probe is still there with our tissue. Then we can easily detect it. So I'll give you some pictures. You just see here. Here, blue colors are representing the cell, but this pink color is the signal. So suppose we have taken the probe with this particular signal. So if this probe is binding with the DNA of this sample, then under certain conditions, it will provide certain UV rays or different things. Different detection procedures are there using which we can get the signal. So if we are getting this pink signal for a particular probe, then we can easily say that this particular mutation is there within this tissue. You just see, these are the signal. These pink color signal is representing that this probe has got their complementary sequences. Okay, so by this procedure, by using probe, we can detect any of the mutated sequence, either in DNA or RNA sequence. So you can see here that Due to this complementary sequences, this probe attaches with the positive one. And if we have got this negative sample, means if the suppose we have got the tissue from the negative sample, means the patient who is not having that particular mutation, then what will happen? Probe will scan the DNA, but probe will not get its complementary sequence. Then what will happen? It will be suspended in the solution. After washing, it will be removed off. When it will be removed off, then what will happen? Under certain conditions, under certain detection procedures, we will not find anything. Means this kind of tissue we will get. Means no pink color signal we will find, because this probe will be washed off. All the probe, since it has not since the probe has not got its own complementary sequences, then it will not ultimately attach to the DNA of that tissue, and it will be washed off, so it will not give any signal. Got it? So by this probes procedure, we can detect any of the genetic disease. So again, it is a nucleic acid based detection procedure. Okay. So something has been written here. We will see what has been written. So single stranded DNA RNA. Either probe is either DNA or RNA, but it should be single stranded. If this would be single stranded, then only it will adhere to its complementary sequence. Okay, to the pathogen. If suppose if we are going to detect any pathogen, then we will have to make the complementary sequence of DNA or RNA based on the DNA sequence of that pathogen. And if we want to detect any of the mutation in our DNA, then using the probe of the complementary sequence of that particular mutation, we can do it. So by using probe, we can detect any of the pathogen, or within our DNA, if we want to detect any of the mutation, we can do it. So its application is for both, to detect any of the pathogen either present in our tissue or any of the mutated gene. So what we are doing, we will make the single stranded DNA or RNA that should be complementary to the pathogen, either pathogen or mutation, means mutated gene. In case of genetic disease, if any of the genetic disease is there, so have been used. Okay. So these probes are also tagged with signal molecule. How we will detect that probe has got its complementary sequence? Based on their signal molecule. So what we will do, we will attach the signal molecule on the probe, using which we can detect either that probe is there with our tissue or washed off. Okay. In positive samples, they form complementary sequence and get hybridized. Hybridized means binding of both the sequences will be called as hybridized. Okay, and give signal. So what will happen when this probe will get its complementary sequence? Then it will hybridize with your DNA, it will hybridize with the pathogen, and after that what will happen? It will start giving the signal. Wherever this probe will be there, it will produce some certain signals, certain signal it will start producing. After that, we can detect that either this probe is there or not. However, in negative samples, they can't bind with the target, okay, due to absence of pathogen or mutation. So if no pathogen is there, if no particular mutation is there, then this probe will not be able to bind with it. And after that, what will happen? We will do the washing procedure, and this probe will be washed off, and it will not be able to provide any signal. So after washing, probes will be washed off and no signal will be detected. It means if signal is not there, it means probe is not there, it means that particular sequence is not there. If signal is there, it means probe is there, it means probe has hybridized with the DNA or RNA with the pathogen or any of the mutation, it means that particular sequence is there. Now we'll come on the next topic. That one is transgenic animal. Okay, so what is transgenic animal? Transgenic has this word transgenic has been formed with two particular words, trans and gene. Trans means other than. Okay, means if we are taking the DNA from another species, from another source, and we are putting the gene from another species, from another source, in an animal, then the animal which has been produced will be called as transgenic animal. It means suppose we have taken the gene from the plant, and if we are putting that gene in animal and we are expressing it, then that animal will be called as transgenic animal. Suppose we have got a gene from insect, and if we are putting that particular gene in animal and we are expressing that, then that animal will be called as transgenic animal. Suppose, and even though if we are taking the gene from another species of the animal, then also that animal will be called as transgenic animal. Suppose we have taken a human gene and that gene we have put in the mice, then again that animal, that mice will be called as transgenic animal, transgenic mice. So transgene means if we are taking gene from different source, different species, and putting in the animal, and we are making a different kind of animal that is producing a particular different type of protein, then that animal will be called a transgenic animal. So I'll show you the transgenic animal with this example. So this one is a goat. You just see here, we have taken the DNA from human. So we have taken this gene. We have taken this gene from the animal, and by this micro injection, I told you before how we are producing the transgenic animal by micro injection method. So we have taken the egg from this donor, we have taken the egg from this goat, and we have taken the gene from the human being, and we are injecting here in this egg. And when we are producing this goat with this particular gene, then this goat will be called as transgenic goat or transgenic animal. Now, another example, you just see here. I told you before what is the microinjection procedure. So we have, you just see, we have taken this fertilized egg. Okay, so you just see here, male pronucleus is there, female pronucleus is there. Before fertilization, we have taken this oocyte. Okay, so in this oocyte before fertilization means here male and female nucleus is there, and it is about to be fused. And before that, what we have done microinjection. So we have taken this fertilized egg, and after that we have molded this one with the microinjection method, and with the needle. What we are doing, we are injecting the DNA. You just see here, we are injecting the DNA, and this DNA comes from another species, either human or another animal, from rabbit or guinea pig or goat or buffalo or any of the plant or insect, from anywhere. If we have taken DNA from another source, another species, then what we are doing, we are injecting the DNA. And here you just see, due to presence of this foreign DNA in this rat or mice, it has become transgenic mice or transgenic rat. Okay, so you just see here. So transgenic animal is one that carries a foreign gene. Okay, so if we are putting any of the gene means from the another source in the animal, any of the foreign gene, if we are putting in the animal, that has been deliberately inserted into its genome. So if we are inserting, or if we are putting any of the foreign gene in any animal, then that animal will be called as transgenic animal. First, transgenesis is the procedure of making transgenic animal or transgenic plant or transgenic organism. Okay, so transgenesis means what? Transgenesis is the procedure using which we are making transgenic organism. So here transgenesis is the process by which mixing up of a gene takes place. So you obviously, we are taking the genes from two different sources and mixing them. That's why this procedure will be called as transgenesis. Now, transgenic technology has led the development of fishes, livestock, and other animals with altered genetic profiles, which are useful to mankind. It means what we are doing, still we are doing all these experiments. Means what we are doing, we are taking the DNA from different sources, and we are making different transgenic animals. We are making transgenic fishes, we are making transgenic livestock species, we are making different organisms to get different phenotype and protein. Suppose if we want to get a human protein in mice, then we can do it by producing transgenic animal. If we want to produce any of the plant protein in the animal, we can do it. Okay, so we are using fish, we are using livestock species. An example I'll take, if we want to make the transgenic goat, which will have the hyper muscle, then what we will do, we will manipulate some genes, we will take the genes from another source, from for the different phenotype, and we can put it and we can make the transgenic animal. So for the mankind, we can produce different transgenic animals from different species. Okay, now till date rat, rabbit, pigs, sheep, cow, fish has been produced. So all these organisms have been produced as a transgenic animal by different by inserting different genes from different organism to get different protein or different phenotype, including mice. And here it has been written that it accounts, means only mice accounts 95% of all the existing transgenic animals. Means whatever transgenic animal we have produced till date, around 90% of the transgenic animal has been produced only mice only. Means out of all these species, only mice contributes around 95% of the transgenic animal. Okay, because it is very much easy to handle the mice, and it is very much easy to generate transgenic mice as compared to the rat, rabbit, pig, sheep, cow, and fishes also. Okay, but although we have produced rat, rabbit, pig, sheep, cow, and fish transgenic organism. Now we'll see what is the use of all these transgenic animals to study the physiology and development. Okay, so if we want to see that what would be the effect of this particular gene, then what we will do? We will put that particular gene in mice. We will put that particular gene in monkey. We will put that particular gene in different species, and we will see the particular role of that particular gene. Either we can overexpress that gene, or we can delete the gene. Suppose I have found a gene. Suppose we are talking about the ADA gene, and if we want to know that what is the function of this ADA gene in immunity, then what we will do? Either we will delete that particular gene in the transgenic mice. Either what we will do? Either we will overexpress that gene. Means what we will do? We will put an extra ADA gene in that particular transgenic mice, and we will see what is going on with this particular mice. So to study the regulation of a gene, how it is regulated, and to assess the functional role of in physiology or metabolism, transgenic animal has been produced. So what we will do? We will produce the transgenic animal with the altered expression. Either we will delete the expression of that particular gene, either we will overexpress the gene. Then we will see as compared to normal what is going on. Suppose we have taken a transgenic mice with deleted ADA gene. Then as compared to normal, we will see what is the physiological role, what is its behavior, what is the altered physiological process in that particular organism. Since we know that ADA gene is playing a key role in immunity, then we will see that in the transgenic animal in which we have deleted that particular gene will show lesser immunity as compared to normal one. So by altered expression of the gene in the transgenic animal, we can get the information what is the role of that particular gene in a particular physiological process. Okay, by manipulating in a particular gene, we can study the effect of altered functional role. So if we can down regulate the expression of that gene or overexpress the particular gene, then we can get the information that how it is playing a particular role in a particular physiological process. Then we can get the information that this gene is playing a key role in this particular physiological process. Okay, suppose I want to see that what is the role of insulin gene, then what we will do? Either I will overexpress this gene in the transgenic animal, or either I will delete it. If I will delete this insulin gene in the transgenic animal, then what will happen? That animal will become diabetic. Then we can say that the for the progression of diabetes, this insulin is very much essential. Means what will happen when this gene will not be fully functional, then only diabetes occurs. We can get this information. So what we are doing, we are manipulating in a particular gene, and we can study the effect of the altered function. So as compared to normal, we will see what is its effect. Okay, the alteration could be overexpression. Means when we are increasing the expression of that particular gene, as compared to normal, then it will be called as overexpression. So I told you that if we are overexpressing this gene, then we can see what is its role, what is the behavior of that particular gene as compared to normal. Either what we are doing, either we are not we are doing knockdown experiment. Means what we will knock down means down regulate the gene. So as compared to normal, if we are suppressing the expression of that particular gene, then again by this altered expression of gene, what is the behavior of that particular gene in that particular transgenic animal, we can do it. Either we can completely delete it, we can completely delete that particular gene in that particular transgenic animal. That is called as knockout. In case of knockout organism or knockout transgenic animal, we can see that if we are completely deleting this gene, what is its role in the normal physiological process. Then we will compare with a normal animal. Then we can identify the role of that particular gene in the normal behavioral process, normal physiological process. Okay. Now, so first we have seen that how we are using the transgenic animal. So first application of this transgenic animal is to study the physiology and development. First thing, and the second application of this transgenic animal is study of the disease. Okay, so I told you before also that if we are deleting any gene, if we are manipulating any gene, then we can see that this gene, due to absence of this gene, which disease is being progressed due to overexpression of this gene, it causes which disease. So by the manipulation of these genes, by the manipulation of the expression of all these genes, we can identify that what is the role of this gene in a particular disease. So here we will see transgenic animals are designed in such a way as we can understand how a particular gene contribute to the development of disease. The transgenic animal of the human disease model can be utilized in investigation of different treatment methods. For example, animal models of cancer, cystic fibrosis, rheumatoid arthritis, etc. have been made. So suppose if we want to see that what would be the corona infection in cancerous patient, then we can't do the same experiment in the human being. Then what we can do, we can produce the cancer model of these transgenic animals. So what we will do, we will produce transgenic mice with a particular cancer. Suppose we want to check what will be the effect of coronavirus in the patients with lung cancer. Then what we will do, we will produce transgenic mice with with the lung cancer, and we will challenge it with the coronavirus. Then we will see what is its effect. If we want to see what is the role of a particular drug in this particular disease, then what we will do, we should have a particular model for that. Suppose anyone is saying this drug is working best in case of this particular disease, then we cannot make a trial in case of human being. Then we should have a model. We should have a model of that particular disease on which we are going to employ that particular medicine. So those data will be generated on animal models. So if we want to study a particular disease, then we should have the model of that particular disease for the human being. And that those models, I told you, could be of mice, could be of rabbit, could be of rat, could be of guinea pig, could be of monkey too. So what we will do, we will manipulate the gene, we will produce that particular disease, and we will see the effect of that particular medicine. Suppose anyone is saying that I have made this vaccine for against the coronavirus. So we cannot, how we will come to know that this would be the dose of this vaccine? What would be the gap in between two doses of this vaccine? So we will make some models, human model, and what we will do, human model or disease model of the human being. Then only we can check the amount of vaccine. We can check the gap of both the vaccinations. So for those things, we need to have some disease model. So we can make the disease model by these transgenic animals. Now we will see the third application for biological product production. Means, suppose I told you before also, we are using bacteria to produce the insulin gene, but not all the protein can be produced in the bacterial gene, because after, you know that protein is present in tertiary and quaternary structure too, that will not be formed in the bacterial system. So some of the proteins can only be made in the animal system only. So what we are doing, suppose we want to make a particular protein, okay, that particular protein can be expressed in bacteria, but it may happen that that would not be fully functional, because no secondary and tertiary and quaternary structure formation takes place in case of bacteria. So what we will do, we will put that particular gene in mice, and we can get the production of that particular gene, that we can get the production of the protein in that particular animal, a transgenic animal. So transgenic animals produce protein for the treatment of several human diseases. Okay, so if we want to produce a particular protein in any of the animal, then we can do it by producing transgenic animal, by putting that particular gene. So transgenic animals produce protein for the treatment of several human diseases. These animals have been created by introduction of a particular gene. Okay, so what we will do, if we want to get a protein of a particular DNA, then that gene of interest we will put in that particular transgenic animal, which codes for a particular protein. Suppose here example has been given, alpha 1 antitrypsin to treat emphysema. So emphysema is a disease in which this particular protein will be utilized. So for this particular protein, what we will do, alpha 1 antitrypsin is a protein for which there will be some gene. So that gene we will put in transgenic animal, and that transgenic animal will start producing this particular protein, and we will collect the protein from the mice or rabbit or any of the transgenic animal, and we will take this protein as a treatment molecule to treat this disease emphysema. So first transgenic cow was produced in 1997, has been produced having milk with enriched alpha lactalbumin. So in 1997, a first transgenic cow was produced, which was producing alpha lactalbumin as to provide the balanced diet for the human babies. So if we want to get a particular protein, if we want to get a particular product, then what we will do, we will have to get information that this gene is producing this particular protein. Then that gene of interest we will use to make the recombinant DNA. By after making that recombinant DNA, we will make the transgenic animal, and that transgenic animal will start producing that particular material. Okay, I told you before that before, and in class only I told you, we have successfully produced the golden rice. So in rice, no vitamin A is being produced naturally. So what we have done, we have used a gene that functions as a precursor of vitamin A. So what we have done, we have put the gene in the rice, then those rice started producing the precursor of vitamin A that is not naturally present in case of rice. So if we are eating the rice, then we are, along with the rice, along with the carbohydrate, along with the food, we are getting vitamin A also. If we want to get any of the product in any of the organism by production of these transgenic technology, transgenic rice, transgenic animal, transgenic plant, we can get any of the phenotype or any of the product. Now, vaccine safety is the fourth application of the transgenic animal. So here, transgenic animals have been utilized as a human model for vaccine testing for polio vaccine. Okay, suppose if anybody is saying that this polio vaccine is being injected in this particular diseased person and that person is dying, then we should have that particular model. We cannot do the experiment as a human being. So what we will do, we will make the model of that particular disease. and we will inject the polio vaccine. After that, we will see what is the effect. So in order to assess the vaccine safety in a particular disease, what we are doing, we are producing transgenic animals, and we are using those transgenic animals as a human model for a particular disease for vaccine testing. Okay. Now, the last application of this transgenic animal is chemical safety testing. Suppose if we are providing any drug, any of the medicine to the person, and if the person is saying that this would be the if the person doesn't know that what would be the limit of the dose, what would be the limit of the weight and the gap and the time of giving these medicines. After a certain time, it will become, after a certain limit, it will become the toxic. So what will happen, we can make the transgenic animal for the human being, and we can assess its toxicity. After a certain dose, if the animal is dying, it means after that particular amount, within that particular dose, the person may die. So for the adjustment of the amount, for the adjustment of the dose, for the adjustment of the days, we can use the transgenic animals. So here, chemical safety testing we are doing with the transgenic animals. So toxicity or safety testing is also being done for drugs testing before starting the human trial. So before human trial, what we are doing, we are taking transgenic animals and we are assessing the dose, we are assessing the amount, we are assessing the time. So all these things we are assessing on the human model, and these human models are nothing but just a transgenic animals. So in this case, transgenic animals are made by the gene manipulations to make more sensitive animals towards the toxicity of the drug. So we are manipulating some genes, we are manipulating some of the genes to make different diseased human model, to make sensitive human being. So what we are doing, we are producing transgenic animal, and on the population of those transgenic animals, we are testing different medicines, we are testing different drugs. So in from next class we will start this ethical issues.